Look at us in this beautiful room today because our other space is co-opted, so we'll take it. Got the mountain views. I'm happy to introduce Dr. Chris Smith. Chris is a postdoc in CIRES and has a background in computational biology. He did his PhD in the Ecology and Evolutionary Biodepartment here at CU, and he studied approximate Bayesian computation methods for demographic inference and evolutionary genetics of sunflowers. My favorite flower. He was also a postdoc at the University of Oregon, where he worked on machine learning approaches for spatial population genetics, and he is very excited about machine learning methods development for ecology and environmental science applications. So today we're going to hear his talk titled Spatial Demographic Inference Using Genomics and Machine Learning. Thank you, Chris.
Thanks, Allison. Hi, everyone. I'm Chris Smith. I'm a postdoc in CIRES, as you just heard. It's my first time here. And I was interested in doing this because I'm trying to network a bit. I'm very excited about environmental data sciences. And I like what you all do in Earth Lab. In particular, I'd like to get involved however I can. So I'm planning to come to these seminars, and I'm going to look for opportunities to collaborate. So thanks for letting me present today. My presentation is about genomics applications in ecology. This is my recent research area. I realize it's not the central focus of environmental data sciences, so I have an intro that reviews some of the methods, some of the different data types in genetics and different applications for genetics, for conservation biology. So I've organized my slides into 3 parts. That was what I was going to say on this slide. So about me, you heard some of this. I went to grad school here at CU, studying evolution and population genetics. And I recently moved back from a postdoc at University of Oregon, working on methods development, also in population genetics. This is what I'm talking about today. And I'm currently working on a greenhouse gas modeling study. Okay, talks organized into 3 parts. This overview of genetics applications I mentioned. This will set the stage for where my research fits in, developing new inference methods. And last, I will share some half-baked ideas for next steps. So jumping into the overview of genetics applications. So biodiversity provides ecosystem services that all life on Earth depends on. Things like pollination, waste decomposition, carbon sequestration, but we're losing species at a fast rate due to habitat loss and other anthropomorphic impacts. 28% of assessed species are currently threatened, so it's never been more urgent to study biodiversity and how to conserve it. So how do we go about conserving biodiversity? Well, there's a ton of work to do here. One important layer that I'm focusing on in this talk is population monitoring. So, for example, conservation biologists need to know how large a population is. Smaller populations are more likely to go extinct. We need to know where individuals are in space, and we need to know whether individuals move between fragmented habitats, because a smaller population is more likely to die out. And we need other data on movement as well. Like we need to know if our animals disperse from their parents. This is important for predicting future range shifts and whether they will keep pace with ongoing climate change. So biologists need these parameters for guiding limited conservation resources. Ideally, we would monitor these things continually in lots of different species, right? Therefore, we need efficient population monitoring techniques. So I've been interested in using genomics for population monitoring. In fact, this is what got me interested in science originally. First interested in research. This was mind-blowing to me that we can go into the field and collect relatively few specimens and then sequence their DNA to unlock this Z-axis. This is all the information contained in the genome. There are billions of base pairs in the genome. This is a ton of data. So as environmental data scientists, I think we should be interested in this source of information. We could do things like compare mutations between genomes to see which ones are more closely related. In turn, we can use that to study movement and connectivity across the landscape. And that last point about movement is something that other technologies like drones and remote sensing doesn't quite give us. And compared to direct measurements like marking and recapturing individuals, to measure movement or to estimate population size. This requires large sample sizes. This is good. There are places for this, however, and radio tracking collars. These are very useful in some situations, but we can't use them on all species, like plants in particular. And these require lots of field work, so extra resources. With genomics, there's potential to use smaller sample sizes, and there's potential for doing non-invasive sampling techniques like collecting hair or poop. What do you think will be easier, tranquilizing wolves from the helicopter, attaching radio collars, or collecting their scat, right? So that's sort of my sales pitch for genomics, for using genomics in conservation. But next I want to dig into this more and discuss the limitations and really evaluate the potential of genomics for doing large-scale biodiversity monitoring. Before we continue, I wanted to introduce some different types of genetic data. I'll have one slide on environmental DNA. With this, we look at one gene or locus. I also won't discuss microsatellites much, but these are traditionally the most commonly used data type for wildlife monitoring and such. And with these, we use several loci or several markers. It's relatively inexpensive to produce a handful of microsatellites, but it's less straightforward to get new ones in new species. I'm going to focus on single nucleotide polymorphisms, SNPs. And when I use the term genomics, I'm thinking of SNPs. SNPs are mutations in a particular site in the genome. These are rows of individuals. So here's a SNP, here's another one, and so forth. With SNPs, we usually work with thousands of markers or up to millions if we spend enough money on it. And the cost of these are continually decreasing as the technology is improving. Think on the order of tens or up to hundreds of dollars per specimen. And with this approach, we generate tons of raw data, and we need a computing cluster to process and distill down the raw data to get these nicely curated SNPs. Okay, here I'm going to showcase some different applications of genetics and conservation biology. And this isn't meant to be comprehensive. These are just some newer approaches that I'm excited to talk about. eDNA itself is not new, but using it for species distribution modeling is recently gaining momentum, to my understanding. And the way this works is we go into the field and collect environmental samples, so water, soil, air, or other things. People are starting to use drones for this, which is rad. This makes all the sense in the world to me because it's high throughput, potentially automated. And then we amplify a particular gene, which identifies the species we're interested in. So then we know where it lives and where it doesn't live across the surveyed area. And that could be used to continually monitor a species distribution. You can also plug this into a logistic regression to study which environmental variables affect survival of our species. And then you can extrapolate to other areas to predict habitat suitability. I think this is an extremely compelling use of genetics, and I predict it's here to stay. It leaves more to be desired, though, because it doesn't tell us about movement or migration, interbreeding between subpopulations. That's something that multi-locus approaches can be useful for. So in my view, the state-of-the-art landscape connectivity analysis program is this one called EAMS. And it uses microsatellites or SNPs to study gene flow across space. So this is tremendously useful for looking at which subpopulations are interbreeding, that kind of thing. There's room for improvement here as well. So the values that EAMS estimates are effective migration rates. And this is a combination of movement and population density. In other words, this red, for example, could be reduced movement or smaller population size. And it seems like in lots of situations, we would want to disentangle those two parameters. Also, these are relative values. So if we want it doesn't give us the magnitude of migration as individuals per generation, which also seems useful. Next is population size estimation, which the field of population genetics has been interested in this for decades. There's too many methods to list, but this one is an example, MSMC. It uses very high quality genomic resources, so like human data, to estimate historical population sizes going back in time to about 100,000 years ago and as recent as several hundred years ago. These years depend on the generation time of the organism, so this is like 20 or 30 generations ago. So as incredible as this is, there are barriers to applying this in a conservation setting. First, these are historical population sizes. It's challenging to get these very recent population sizes. And in conservation, we usually want to know the population size today and like how it's changed since last field season, right? Second point, this model and related models are not spatial. They assume the modeled population is randomly mating, like a jar of flies, so you can't really apply it to study heterogeneous density across the species range. For that, we would need spatially explicit models. Okay, I have a whole slide dedicated to this simulation program, SLIM, which is a cool new data sciencey tool that folks in ecology and evolution are excited about because it can incorporate tons of biological detail and realism in the simulation. In fact, there's a 1,000-page manual for SLIM. It can do things like geography, interactions between species. You can give it input maps, environmental rasters, and tell it how the organism interacts with the environment. And it does all this while tracking the genomes of all the individuals. So there's a lot we can do with this program. One limitation is speed. We can currently simulate moderate populations, but not large populations, I would say. So that was the overview of genetics applications, and now transitioning to my research, developing new inference methods. I hope you're amped on genetics now. So the aim of this project is to explore whether deep learning can help us infer some of these parameters I've been talking about. And deep learning models have outperformed traditional statistical models in a growing number of cases. And so I'm excited about exploring the potential of deep learning here, and in general, I'm excited about machine learning. One constraint with deep learning is it requires very large training sets. And in most species, we don't have abundant data. So I'm going to rely on simulations for this. This movie is showing one of my simulations, and what you see are offspring dispersing from their parents each generation. This doesn't look realistic, I realize. It's a very simple model for benchmark and new inference method. But this uses that SLIM program I mentioned, so you can customize it and make it more realistic. It's a population genetic simulator, so we get SNPs out of this. And then, so I'm going to use these SNPs as input to a neural network to estimate dispersal rate. So these are the dispersal rates that went into each simulation. It's an inverse inference problem. And to clarify what I mean by dispersal rate, specifically I'm estimating this sigma parameter from the literature. This is related to the distance offspring move from their parents. Here I'm not yet thinking about how clumped or uniformly distributed individuals are, and I'm not thinking about range expansions here. The kind of neural network I'm using is a convolutional neural network, or CNN. I know some of you are familiar with this, but CNNs are really good at image-related tasks, and it works by striding a small window or kernel over the image, which can recognize local features that are important to the task. So it might recognize the trunk for classifying this as a picture of an elephant. And researchers have had this cool idea that DNA sequence alignments also look like images from the perspective of a CNN, so we can use the same technology to extract useful information from DNA. So I built a CNN for estimating dispersal rate, and this is the architecture. I was going to walk you through this. It's hard to see this leftmost thing, but this is a big table of SNPs, and this green box is the convolution kernel. It covers 2 SNPs, so we get some information about linkage between SNPs. And it's a 1D convolution that strides across SNPs. We use several iterations of convolution and pooling, and we show it one more piece of information, which is the width of the sampling area. And that's important for when the training examples have different widths. So in some ways, this is a basic CNN, a vanilla neural network, but something interesting about this application is we're using summary information from the entire sample. instead of individuals, we don't even show it these individual sampling coordinates. I'm going to come back to that idea in a little bit. But I validated our new method on simulated data. X-axis is the true dispersal rate from the simulation, and this is the prediction. Sample size is 10 individuals. This calculation is the mean relative absolute error. And so our method is outperforming these other two. In addition, this method requires extra information. It requires an independent estimate for population density. So it really limits the usefulness of this one because in most populations where we're studying dispersal, we also don't know population density. This method also requires extra information. It uses very high quality genomic resources called identity by descent blocks. So these are very expensive to produce. We don't have them for many species. SNPs are much more accessible. So even when we give these methods, these idealistic extra pieces of information, ours is still performing better. But there's a limitation with dispersion as well, which is, well, as some of you know, deep learning models can struggle to generalize to out-of-sample test data. So in this experiment, the test data looked just like the training data. So next, I'm going to check how well dispersion performs with out-of-sample data. So what's going on here is I'm going to tweak a parameter of the simulation, population density. Previously, the training examples all shared the same density. Here, I'm applying that model to test simulations with different densities, and this messes things up. It doesn't perform well in that scenario. So next, I try something new. I trained a new model on these data, which each have different densities. So I'm exploring a range of densities, and then we regain that good performance from before without knowing population density in the test. This in particular is an important finding because that Rouset method from the previous slide can't do this, estimating dispersal without knowing density. Okay, so next I applied this model to very large populations, and here the outputs are biased. So we weren't able to extrapolate to extreme cases. Okay, next I'm going to tweak other parameters in the simulation model. So population size history, the size of the habitat, and the width of the sampling area. So in each of these cases, I varied this nuisance parameter among the training examples to explore a range of values. And this lets us accurately estimate dispersal rate. In these two examples, we couldn't extrapolate to extreme cases, but down here we were able to extrapolate somewhat, so that's neat. So to recap, we were able to address each of these unknown parameters using this procedure I described, sort of sweep them under the rug, at least individually, but a separate question is, can we train one big model that can be used on lots of different species, a foundation model, as it's called in environmental science? So I took a crack at this. So here I'm training a new model, and I'm varying each of these parameters simultaneously. So in other words, if we take a random simulation from the training set, it has a random dispersal rate, that's our parameter of interest. It also has a random population density, demographic history, habitat width, sampling area, plus a random sample size. And that last point will let us apply this to data sets of different sizes. Okay, here we're evaluating the foundation model on held out simulations produced using the same distribution from training. And the axes are log transformed, log scaled now. They weren't in the previous slide. And the error is larger. So this means that predictions were 55% off from the true value on average. So the foundation model isn't super accurate, but when we zoom out across orders and look across orders of magnitude, there's this decently good, interesting signal. So we applied the foundation model to a dozen different species, and we got somewhat reasonable estimates for some of these. The flying animals had larger dispersal rates. The plants had smaller dispersal rates on average. There were questionable ones here, like mosquito. The estimate for sigma was 10 kilometers. Most mosquitoes don't fly that far. This could, reflect occasional long distance migration events. But in any case, I think this served to demonstrate the new method. In practice, I don't know how useful the foundation model will be. I'm suggesting for researchers to not use the foundation model, but to train a new model, customize the simulations to their study organism, make it more realistic, and then they can trust the output better. And I've been corresponding with folks who are applying this to corals, feral pig, Florida scrub jay, for example. So next I had an idea for a new architecture that does use the geographic sampling coordinates. The previous model didn't use these. And we're still estimating dispersal rate. This model outperforms the previous one. I'm going to zoom in on the architecture here. So what's new about this model is it's focusing on pairs of individuals. Specifically, I wanted to look at the relatedness, genetic relatedness between me, that guy way over there, relative to the geographic distance between us. That's the information I think is important for estimating dispersal. And so what we do is subset the SNPs for one pair. So there's two columns, apply convolution as before, and then we show it the latitude and longitude for this pair so it gets to see the geographic distance and the genetic relatedness between this pair, and then repeat that procedure for all pairs in the sample, stack together the outputs before calculating sigma. So this worked really good. It helped us outperform the previous model. And this pairwise convolution thing, it hasn't been done before in genetics or anywhere else to my knowledge. So I was going to share other details about this. So the model weights in this first part of the network that gets repeated, these weights are shared across pairs. So model complexity stays low, even with larger sample sizes. But with larger samples, we do start to run out of memory on the GPU. And so to address that, I exclude most pairs from the calculation of the gradient here. So only a subset of pairs are used to optimize these weights, and that saves a ton of memory. Meanwhile, all pairs are still included in the forward pass here. So this lets us do more with limited memory on the GPU. And we released a Python pip package for this new model. All right, so far I've talked about estimating just one dispersal rate for the whole population. But we know that real environments are not flat and uniform, they're heterogeneous. There are barriers to dispersal, non-uniform resources. So next I wanted to estimate maps. And this is, so I came up with a model for doing this. It uses the same inputs as before, SNPs, geographic coordinates. But here we're estimating two outputs simultaneously. One is a geographic map of local population density, and a separate geographic map showing dispersal rate as it varies across space. And zooming in on the architecture here. So the way this model works is to estimate the value of a grid cell, I wanted to use information from all pairs because that maximizes the use of available data, but not all pairs are near the grid cell. Some are far away, those seem less important. The ones near the grid cell seem more important. So my first thought was to weight the contribution of each pair based on the distance to the grid cell. But the next thought is, what if a pair is on either side of the grid cell? That pair does seem important for estimating dispersal rate because that cell controls movement between them. So instead of relying on the Euclidean distance, I told the neural network to figure out the most appropriate spatial weights for doing this reweighting. And that's what this second input branch is doing. It processes this table of geographic information and it determines spatial weights that get multiplied by the genetic information in order to reweight the contribution of each pair to each pixel. Okay, so this works nicely, and we're going to again evaluate this on simulations. The simulation looks different now. We're using these input maps. There's two maps here, red and blue. The magnitude of red conveys local carrying capacity that controls population density. And the magnitude of blue conveys the dispersal rate in each pixel of the map. So there's this uniform red map, like you saw. In the dispersal map, there's this gap, so guys can't disperse across. And in this iteration of the project, we're using very coarse spatial maps with coarse segmentation like this, so that will affect the resolution of the inference in the end. Okay, so here we're testing the new program Mapin on an example simulation. These are the true maps on the far left. The dispersal map is up top, and so what these values mean are in each grid cell I tracked dispersal throughout the simulation. So these are the actual realized dispersal rates, and here I tracked population density in each grid cell during the simulation. These are the true maps, and then we apply Mapin to the genotypes from the simulation to predict maps. And we're doing decently good here at recapitulating the spatial breaks. Like here, you can see it pretty good. And then these are two other methods that do similar things. So themes is very similar to EAMS, which I introduced earlier. It's estimating a map of effective migration, which is a combination of dispersal and population density. And you can kind of see that in the output. So it's a bit of an apples to oranges comparison. It's not doing the same thing as us. In some ways, our task is more challenging because it's two different maps. This third program, MAPS, it does estimate separate maps for migration and local population density, but it doesn't do well with moderate numbers of SNPs. We're only giving it 5,000 SNPs here. This one also requires identity by descent blocks. I mentioned that earlier for a different program, which we simply don't have for many species. So Mapin is addressing this gap where we want to use SNPs for estimating two different maps, one for dispersal and one for density. Okay, so here again, checked if how well Mapin performs out of sample test data. And so, cutting the chase here, when we tested on extreme cases, the output was biased. It was restricted to this edge of the training distribution. So we weren't able to extrapolate to extreme values. And so to implement this approach, we need to make sure the training distribution covers the true value, sort of the lesson. I demonstrated Mapin on published data in North American gray wolves. So here the simulation looks a little different again. Now it's this cookie-cutted outline. The habitat is this outline of Alaska here and Canada. And then these are the estimates that come out in the end, applying it to real data. Top left is dispersal, and our output showed higher dispersal rates in northern Canada, which is corroborated by data on territory size, which is correlated with latitude. So that's neat. But our density map showed higher wolf numbers in eastern Canada, which isn't quite right. And also, we have an uncertainty analysis using parametric bootstrapping. This showed higher uncertainty in eastern Canada as well. So this appears to be an error. And I think it's due to a lack of sampling in that region. So we're still working out the kinks with this approach. So looping back to our question from before, can deep learning help us infer spatial demographic information? So our new approach, Mapin, uses SNPs. These are accessible input data. We have these for some endangered species now. Uses SNPs to estimate the magnitude of dispersal across space and a separate map for population density. There are limitations that I've mentioned. Our approach struggles to generalize to out-of-sample tests. And since it's trained on simulations, it's probably going to struggle to generalize to real data as well. But I'm excited about this research area, and it's still in the very early stages of development. Okay, with my remaining few slides, I was going to propose some future research directions. And okay, crazy idea number one. I'm interested in collecting all of the published spatial genomic data sets and sort of harmonizing them to create a proper training set for deep learning. So taking a step back, the motivation here is that most research labs study a particular organism and they sequence DNA in that organism, and they're kind of working in a vacuum, so to speak. And so I'm curious about what we might gain by combining all of those separate data sets from these different species. I estimate there are on the order of hundreds of good data sets available for this. That number's growing all the time. When we plug the word SNPs into Dryad, we get 2,000 hits. Some of these aren't spatial. I think the number is closer to hundreds. At first glance, this might not seem big enough to do deep learning, but we can augment the training set by subsetting these individual data sets into many smaller data sets by subsampling specimens or chopping the genome up into pieces and subsampling SNPs or genomic regions. Okay, the next challenge here is we don't know the true dispersal rate for these species. We don't have labels for these training examples, but we might use some machine learning tricks to still make use of these data. So I'm thinking of domain adaptation with a gradient reversal layer. This will let us train primarily on simulations while also showing it examples of real data during training. And then it learns to identify useful features that are shared between the simulated and real data. So what does this mean for conservation? My hope is that by training on real data with domain adaptation, it'll help us generalize to real data. It'll address that shortcoming. And in any case, I think this harmonized data product would be useful for something like in evolution or biodiversity science more generally. Next, I'm interested in combining environmental data with genomic data. And people are doing this in various ways. But I had some ideas to build upon this map-in framework I've been talking about. So we can use environmental rasters to inform the training maps for mapping. This is a sampling of the training maps I'm using currently. And these don't look real at all, right? So in the first scenario, we don't know how this environmental variable affects our species. But Peter Ralph had this idea that we might still make use of this information indirectly. We could just take elevation, an arbitrary variable, and rescale these values to a random range. And that's one of our training maps for carrying capacity. So it's just, the aim there is just making the training maps look a bit more natural. That's an easy way to incorporate environmental data. And then in the next scenario, ideally, we do know how the environmental variable affects our species. So we could build that into the simulation, make it much more realistic. And so that would improve the inference of the connectivity map, for example. Okay, and then how do we get that information about prior information about how this variable affects our species from distribution modeling? So we could take the output from distribution modeling, habitat suitability scores, multiply it by a random carrying capacity, and that's one of our training maps. So there, we're still estimating the overall population size, but the spatial heterogeneity is determined by the output from distribution modeling. In fact, this is precisely what Nolan Kane's lab in eBio is doing at CU. In orchids, his graduate student Jacqueline Matos is leading that. And so because of this, I've gotten really interested in distribution modeling now. And then there's this like dovetailing of interests because I was already getting into satellite imagery from my current postdoc working on methane. People are using satellites for distribution modeling. So I've started to read about machine learning approaches for satellite imagery as well. Okay, last idea is thinking about landscape genetics. So this is using environmental data to predict features of genetic variation, so genetic summary statistics. And a recent development in this area used satellite imagery to predict these two different metrics, allelic diversity and genetic differentiation. So to make this useful for conservation, we would need to do some subsequent modeling using these data. But I thought this was another exciting combination of genetics and satellite data. Okay, we've reached the end, and this work was done in the Kern-Ralph Collab and funded by an NIH postdoc fellowship. Thank you all so much. Happy to take questions. Yeah.
I have two questions, one technical detail and one broad, if that's okay. The
Yeah.
technical detail one, when you line up the SNPs for your CNN, what determines the order of those SNPs? Are they the same each time? Are they randomized between like why are they in order?
Yeah, good question. Thanks. So they are ordered by their position in the genome in the implementation I've done here. But you can imagine like doing different things with that. You could shuffle the SNPs, and that would let you train a model that's appropriate for reduced representation sequencing approaches where you don't have a reference genome. So then you don't know the order. You could do that in the training as well. Yeah.
So if they're going at a pairwise down there, are they not just ranking? It's sort of like if you turn it into like a 1D CNN going down, and they only are seeing 2 at a time. Or not just sort of bubble sorting the SNPs.
I mean, well, it's not sorting SNPs. It's like, oh, here's a pair where there's something noteworthy, you know, output a non-zero value there.
Cool. I'll ask the broad question.
Yeah.
So I know that so there's an example, CSU just cloned fluctuating pair.
Cool.
Did this because there were like 6 natural individuals and there wasn't enough integration population. So they pulled a museum specimen from 100 years ago and they're trying to breed it.
Cool.
This has brought up this question of like, what would be the best time period to pull a sample from if you need to revive a population? So I was just curious, you were looking sort of through space at a lot of the quality of genome fitting with something else. But what if we were looking at the time dimension? Is there something that we could use to say 50-year-old specimen, 300-year-old specimen, best features that we heard?
That sounds really cool. My thought there is the SLIM simulation program. I can totally envision a simulation-based inference framework for like planning that, where you simulate this earlier specimen collection, and then 100 years pass, and then we sequence DNA or whatever, and figure out like what's the optimal amount of genetic variation. I think the SLIM program could be useful for that. Yeah, I can imagine a cool study doing that. Thank you. Yes.
I'm not sure about your like the architecture of your CNNs.
Yeah.
So how did you tune the parameters for the CNNs themselves? Like, how did you decide how many layers to include, the functions that you use? How long did it take to train? And how did you, right? How many did you test out before you decided this was the one?
Yeah, I think I can address that. So like this is the first version. This is the first one, and it took a lot of time developing and iteration with different numbers of layers and sizes and the different dimensions. And so I basically did a grid search of like a bunch of different parameter combinations to see which one works best. And then going forward, I avoided redoing that and sort of just borrowed some of the exact same parameters from before. And like, so here there were some new pieces, and so I tried a few different parameter combinations, different combinations of layer sizes and such. So so in the first model, there's a lot of upfront playing with the numbers of layers like you're describing, and then these later models, I didn't do as much of that. I was thinking more of like, how do we do something totally new with it? What was your
So I'm saying,
The
how
latter
long does it
question?
take for you to train?
So in the examples I showed in the tests, I used 5,000 SNPs and 10 or 100 individuals, and it trains pretty quick, an hour or something on a GPU. But the Wolf one used larger input and trained for weeks, 2 weeks, I think.
So if you had in the end, you know, you're saying that you wanted to use all of that data, how long do you think it would take to train them?
So like, I'm sorry, which,
Yeah, sorry, and you know, you're looking at like future directions of what's to. Yeah, so that, yeah, how long would that be? Would you think that you need more layers in the network to deal with that? I mean.
Nice.
I think that's the biggest, the struggle with not the struggle with deep learning, but you know, it's like, they have better prediction accuracies than like statistical models, but also like they can take so long to train. But once they're trained, you know, you can just plug and go. But I was just curious, like with the data you're working with, what does the training look like in that kind of process?
Yeah, I totally see where you're coming from now. So, you know, the training time is a constraint. Like we can't use some species, we have 1 million SNPs, 1 million loci, and right now the program I have won't work with that. For doing this, for combining all of the real data, this is actually still small compared to the number of simulated data sets I was using. I was using like 50,000. So I don't think that will be the main issue with this particular idea of getting all the real data together. Yeah. Thank you. Great questions.
Yeah, this is super cool. And this is way above my grade for the most part, but I think it was crazy idea number 2. Yeah, this one, where you're talking about taking that like middle panel, like the environmental features and how we know those affect dispersion or dispersal, and then using that to inform models.
Yeah.
I would imagine like on lands, that would be things like steepness of hills or like how much vegetation, or composition of substrate and stuff like that.
Yeah.
Do you think you could maybe do a similar thing going like into the ocean and doing like thinking about how currents are interacting? Because I mean, like marine species are like dispersing in very different ways than species on land for the most part. I'm just curious if you thought about that at all.
Nice. I've superficially thought about these things some. I forgot to mention there's a whole field of landscape genetics that uses environmental data. Oh, well, I mentioned it here, but you could do some modeling to you could use this circuitry modeling to like kind of figure out which of those land variables you mentioned are more important for connectivity. So I can imagine kind of merging, I'm speaking in very vague terms, merging that literature, those ideas with like what I've done here. Thinking about like seascapes, I've corresponded with a guy at University of Oregon studying corals. So like, if you said his name, I would know it. Sorry, I don't remember. But the corals, the habitat follows the coastline, to my understanding, of this archipelago he's looking at. And so he wants to model dispersal, but it must follow the coastline. So it's kind of linear. But the distances I calculate here and the dispersal distances in SLIM simulation are Euclidean distance. But he's like, I don't want it to jump over land. I want to calculate these kind of linear circumferential distances. So where I'm going with this is where some folks are thinking about marine and seascape habitats, and we might need to make some tweaks to the simulation to figure out the best way to do that.
So it's almost like how the ocean's interacting with the landscape. It would be sort of an important driver for it.
Yeah.
Yeah. Thanks.
want to note that online, Joey Taylor says the idea of allowing the spatial weighting to vary to recover more geographic information is really neat. Thanks for presenting.
Oh, thank you.
Okay.
Thanks, everyone.